Hello, everyone, and welcome to today's webinar, Validating a Pharmaceutical Total Organic Carbon Benchtop Analyzer to ICH-Q2. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter. To learn more about our sponsor, please visit www.beckman.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Tony Harrison, Senior Marketing Manager at Beckman Coulter. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Tony, you may now begin your presentation.
Excellent. Thank you very much for that introduction. So, hello everybody. My name's Tony Harrison, and I am a product manager for Beckman Coulter Life Sciences. And today I'm going to talk about validating a pharmaceutical grade TOC laboratory analyzer to the International Committee on Harmonization Procedure, ICH-Q2. Now, ICH-Q2 was written by representatives from the United States, the Japanese, and the European Pharmacopoeia, and it contains guidance on how to ensure that a TOC analyzer is fit for purpose and for providing batch release data. ICH-Q2 has taken on special importance since it was referenced in the FDA's December 2018 guidance on their 21 CFR Part 11 data integrity compliance. A recent report shows that in 2017, almost 75% of the FDA's 483 warning letters cited data integrity issues. And the main focus of these letters was not security of the electronic records themselves, but in fact the accuracy of the records. They are encouraging pharmaceutical industry to improve their testing processes to make them more robust to avoid the natural human error that creeps in during complex QC testing and introduces error to the data records. QC testing instruments can be complex to configure and operate, and it is easy to make mistakes, which is why here at Beckman Coulter we've made our QBD 1200 TOC analyzer simple and straightforward to configure and use. So by making the QBD 1200 TOC analyzer simple to use, we're helping pharmaceutical QC managers to reduce their training burdens. And by allowing the user to program their standard operating procedure, their SOP, into the QBD 1200 and automating it, we ensure that the SOP is followed every time, and again, reducing human errors and avoiding problems with the inaccuracies in the electronic records. Here at Beckman Coulter, we manufacture a range of QC instrumentation that have all been optimized to provide robust QC testing for the pharmaceutical industry. All of them allow the user to program in and automate their standard operating procedures, helping to avoid human errors and reducing the operator required skill level so that the QC manager doesn't need to have highly skilled operators carrying out routine and repetitive QC tests. Our instruments cover a range of parameters from TOC analysis to mammalian cell concentration and health, cleanroom air monitoring, final injectable drug quality testing, and stability testing for protein-based biologics. The QBD 1200 analyzer is designed specifically to suit the pharmaceutical QC application. It can analyze TOC levels from 0.4 ppb to up to 100,000 ppb. It uses the industry standard UV and persulfate method and can handle up to 64 vials in the autosampler at a time. As I mentioned, the pharmacopoeial tests are in there, and you can pre-program your standard operating routine into it. And then you simply select the SOP or the compendial test like system suitability using an intuitive icon-driven menu. So in the analysis, the first step we do is we acidify the water sample. This encourages any total inorganic carbon, which is typically made up from carbon dioxide dissolved in the water, and it gets that total inorganic carbon out of solution and into the water as a gas. And then we use a carrier gas to actually sparge or purge the carbon dioxide from the total inorganic carbon out of solution, where it's measured using a non-dispersive infrared detector, and we report the TIC. The next step is a strong UV light is turned on, and this tries to release the carbon atoms in the organic material present in the sample. The organic material is oxidized by a combination of hydroxyl radicals from the water itself and by persulfate, which in the presence of the UV light forms a very strong oxidizer. And then the TOC is released as carbon dioxide gas. And then the third step is a direct measurement of that TOC. So the carbon dioxide from the TOC is sparged out or swept out from the solution using the purge gas and is measured by the non-dispersive infrared detector. So we wait till all the TIC, the dissolved carbon dioxide from the TIC, is gone. And then we turn the UV lamp on, and that's when we start to oxidize the organics, and then we measure the carbon dioxide from that. So we make sure there's no interference from the TIC. Now we have a documented IQ, OQ, and PQ procedure available. And it's the PQ section we're going to talk about today because that is the one that carries out the required testing to validate the QBD 1200 to the ICH Q2 guidelines. For those of you who are not familiar with it, ICH Q2 is in fact the International Conference on Harmonization. And they're working to harmonize the pharmacopoeias between European, United States, and Japanese. And they also produce guidance such as the ICH Q2 guidance. So what's ICH Q2 itself? Well, there's various parameters that they recommend or attributes that they recommend, and they give guidance on these attributes. And they ensure that they're tested to make sure the instrument is suitable for pharmaceutical testing application. It covers instrument accuracy, precision, specificity, limit of detection and quantitation, linearity, range, and finally, robustness. So let's take a look at each of those attributes in detail and how the PQ for the QBD 1200 demonstrates compliance. Now, ICH Q2 suggests that an instrument's accuracy can be best demonstrated by examining the instrument's response to a certified reference standard. The QBD 1200 PQ uses a certified potassium hydrogen phthalate solution containing 5 parts per million of total organic carbon. Automated onboard dilution allows comparison against this standard at 5 points across the measurement range. And the test SOP is preloaded into the QBD and automated, and it's simply selected from an icon on the screen. Although the United States Pharmacopoeia and the European Pharmacopoeia don't specify the calibration solution to be used on TOC analyzers, the Japanese Pharmacopoeia does. Japanese Pharmacopoeia requires potassium hydrogen phthalate to be used. Hence, by using this, the QBD can be seen to comply completely with all the pharmacopoeias globally. The QBD 1200 accuracy is demonstrated by comparing its response to 5 different levels of TOC from that certified standard. And the pass-fail calculation is automated and avoids manual calculations and human error. ICH Q2 suggests that precision can be assessed by comparing multiple analyses of the same sample. It suggests 3 replicates of 3 different concentrations of TOC, and the PQ from the QBD 1200 follows this guidance exactly by doing 3 replicates each of 1, 2, 3, 4, and 5 ppm concentrations of TOC solution. The performance of the QBD 1200 against this precision test is calculated automatically in a pass-fail generated. A quick word on accuracy and precision. In this diagram, we're explaining the difference between these two. The first dartboard shows what happens when you have neither accuracy nor precision. The results of these repeated tests are all over the place and nowhere near what we'd like to see. The second dartboard shows excellent precision with very repeatable results, but it's not very accurate. It's got poor accuracy but high precision. The third dartboard shows relatively good accuracy in the sense that the results are clustered around the bullseye, but the precision is poor as the results are not consistent. They vary quite a lot. And finally, the last dartboard, that's exactly what we want, where the instrument has got both good accuracy and high precision, with the results on target and reproducible. Now, the specificity test demonstrates that the TOC analyzer can measure and report TOC accurately in the presence of potentially interfering substances that may be present in the sample. Now, this is demonstrated by carrying out 2 tests. One is a system suitability test to ensure that the TOC analyzer can successfully analyze the full range of organic materials expected to be present in pharmaceutical grade water. And secondly, by demonstrating that the analyzer can successfully report TOC in the presence of large amounts of total inorganic carbon in the form of dissolved carbon dioxide. Now, most pharmacopoeias determine that the range of organics likely to be present in the sample can be covered by testing using sucrose and benzoquinone. And this test is preloaded and automated with automatic pass-fail in the system. However, the Japanese pharmacopoeia suggests that specificity can be demonstrated by the successful analysis of SDVS. And again, that test is built into the analyzer and can be selected from a simple button on the screen. The second part of the specificity test that we do on the QBD 1200 is to test it in the presence of very high levels of total inorganic carbon by completely sparging it from the sample before we measure the TOC. So we put a sample in which contains high levels of TIC, and we make sure this doesn't interfere with the measurement. So we don't turn our UV lamp on to start the TOC analysis until we are ready to do that. We've got rid of all the TIC from the sample. So TOC analyzers use 2 separate sensors to measure TIC and then total carbon can suffer badly, in fact, in samples containing large amounts of TIC in the form of dissolved carbon dioxide. And in fact, that's very common to have large amounts of dissolved carbon dioxide, especially in purified water samples. Small errors in each of these sensors, a measurement can actually cause really large offsets in the reported TOC and even lead to negative TOC results. TOC analyzers that use the 2 sensors to calculate the TOC, they calculate it by subtracting the measured total inorganic carbon from the total carbon that they measure. You can see on this screen here is a common TOC analyzer found on the market, and you can see it's actually measuring IC, inorganic carbon that is, and total carbon, and it's calculating the TOC. In this particular case, the purified water contains 12.3 parts per million of TIC, and it's reporting that calculated TOC value is 600 parts per billion. But, however, the sensors that are making these calculations actually have a plus or minus 2% tolerance on the 2 sensors, which means that as these two sensors drift away from each other by plus or minus 2%, the reported values for TIC and TC can vary tremendously. So the actual level of TOC present or reported could be anywhere between 96 parts per billion and 1,104 ppb. The plus or minus 2% tolerance on the QBD single sensor means that when it actually does a measurement, it's actually using the same sensor to make both measurements. So potentially, if you've got 100, say for instance, 100 parts per billion of TOC in your sample, then you'll get a plus or minus 4 ppb maximum error on that sample measurement. Now, the limit of detection is, in fact, the smallest amount of a substance that any analyzer can detect. It might not necessarily accurately report it, though, and that's where limit of quantitation comes in. This is the smallest amount of a substance that the analyzer will not only detect, but will accurately measure and report. Now, the performance qualification follows the ICH Q2 guidance on how to calculate the limit of detection and limit of quantitation. So these are calculated values. It's almost impossible in the field to demonstrate the limit of detection and quantitation because the levels of TOC are so low, you can't get calibration standards that low. So they're calculated. The linearity test determines the ability of an analyzer to report accurately over a range of analyses. The PQ for the QBD 1200 demonstrates this by showing the linear performance of the QBD 1200 across a range of measurements. So we put the TOC standard through and we look at the response from the analyzer for multiple points across the measurement range. And the pass-fail calculation is automated in the analyzer, so there's no manual calculations, and hence we try to eliminate human error. Now, the range of the QBD 1200 is demonstrated by testing in the measurement range and then explaining that higher levels of TOC are analyzed through automatic sample dilution using a calibrated syringe pump accurate to plus or minus 1%. Now, it's important to realize that the TOC, we report it in parts per billion. And actually what that's equivalent to is 1 microgram per liter of water. And of course, a liter of water is 1 kilogram if it's really ultra pure water. So we're reporting 1 microgram in 1 kilogram. Now, it's important to understand that's what we're measuring here, because if you've got, say, a 1 liter sample of water containing 100,000 parts per billion of TOC, if you dilute that with another 1 liter of TOC-free water, the TOC level is halved, because now we've got twice the amount of water but the same amount of carbon. So it now would report 50,000 ppb. So using this, we can now use dilution to allow us to measure higher levels of TOC. In samples of up to 4,000 ppb, the dilution is not required, as the amount of persulfate added automatically each time by the QBD 1200 will successfully analyze this level of TOC. However, above 4,000 parts per billion, the QBD 1200 automatically determines the amount of persulfate to add, so the user does not need to guess. Robustness is the ability to measure successfully despite variations in the sample characteristics. And the way we do demonstrate that with the PQ for the QBD 1200 is by testing the QBD 1200 with samples where the pH has deliberately been altered. So we put the same level of TOC in with different pH values and check that it will still measure despite this variation. Now, intermediate precision is an interesting one. What they're talking about here is the effect of random events on the precision of the analytical procedure. And what they're referencing really here is having different analysts use the machine, and the way they use the machine might give slightly different results. So it gives you a differing result depending on what the technician is and depending on their skill level as well. And the way we actually demonstrate compliance to this with the QBD 1200 is we actually automate the SOP, So the integrant's automatically configured correctly every time, so the user doesn't configure the machine. And also we automate the addition of persulfate to ensure that the TOC analysis is analyzed correctly. Now that might seem obvious, but in some TOC analyzers, particularly those using a carbon dioxide permeable membrane as part of the measurement process, the users have to guess how much persulfate to add. In other words, they need to know the level of TOC present in the sample before they carry out the analysis. But how can they know that? The problem is, you see, if they're using these membrane-based technology and they add too much persulfate for the amount of TOC present, bubbles form in the analysis chamber, and this blocks the carbon dioxide permeable membranes, causing the TOC analyzer to underreport because the carbon dioxide doesn't go through the membrane. Now, equally, if they don't add enough persulfate for the level of TOC in the sample, the TOC will not be fully oxidized, and again, the TOC analyzer will underreport. Finally, the PQ demonstrates its robustness by being unaffected by gas bubbles in the sample. If you remember, I mentioned the membrane permeable ones. If they get bubbles in the sample, it blocks the membrane and the carbon dioxide is not measured correctly. But in this case, we measure the carbon dioxide using a non-dispersive infrared, which is selective to measuring only carbon dioxide. So it doesn't matter if there are bubbles of other gas present. And the standard amount of persulfate we add covers even the widest range of TOC analysis required in pharmaceutical grade water. In other words, up to 4,000 ppb of TOC. And of course, the pass-fail criteria in the pharmacopoeias is 500 ppb. So we should be coping with the maximum amount of TOC that you'll find in your sample. So that brings me to the end of my presentation today. And thank you for listening in. And if you've got any questions, please submit them now.
Thank you, Tony, for your presentation. We will now be pushing out a poll question, which will show up on your screen. We appreciate your participation. Okay, we will now get started with the live Q&A session. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of the presentation window labeled Ask a Question and click on the Send button. Tony will answer as many questions as time permits. All right, let's get started. Our first question is, why is it important to validate a TOC analyzer to ICH-Q2?
Yeah, no, certainly. I mean, most validation packages that you get with an off-the-shelf type of instrumentation, it really validates that the machine works correctly and it does what it's supposed to do. But in fact, if you're using a QC laboratory TOC analyzer to provide data for the batch release file, then it should be validated for that application, and that's where ICH-Q2 comes in. It provides sufficient evidence that's acceptable to the regulatory inspectors from the United States, the European and the Japanese regulatory bodies that the QC lab analyzer is fit for that use.
Great, thank you. Okay, next question. How does having single sensor technology help the QBD-1200 avoid interference from high levels of total inorganic carbon?
Certainly, yeah. So the problem we're trying to avoid really is, in TOC analyzers that use two separate sensors to measure total carbon and total inorganic carbon. And the problem is with two sensors, a small drifting calibration between the sensors can lead to measurement errors which will impact on the accuracy of the calculated TOC, or total organic carbon. And what we do with the QED-1200 is we trap an aliquot of the water sample, we measure the total inorganic carbon by sparging it out with the carrier gas, and then we oxidize the sample to measure TOC with the same sensor. So any drift in the single sensor will affect both the measurement of the total inorganic carbon and the total organic carbon. So it'll affect both measurements and effectively cancels the drift out.
Thank you. How does the QBD 1200 avoid the effects of human error on the intermediate precision as mentioned in ICH-Q2?
Yeah, no, good question. So as I mentioned, the intermediate precision is really looking to see what effect can be made on the measurements between different operators who are configuring the machine slightly differently or configuring the sample or things like that. So what we've done is we simplify the instrument setup. We reduce the configuration steps, so we're not having to decide how much persulfate to add. It adds the correct amount. And that reduces the opportunities for human error. And that human error can occur more regularly in more complex lab TOC analyzers. And so also the benefit of that is we're reducing the need for continuous training to operate the instrument, and for the benefit for the QC managers, we're reducing the skill set that the operator needs. So you're not having a highly trained technicians doing repetitive, mundane QC testing. You can de-skill it effectively and program the SOP and just select it from an icon. So the whole point is by simplifying the instrument, we've reduced the effect of operator-to-operator skill levels and hence reduced the intermediate precision errors as outlined in ICH-Q2.
I would like to once again thank Tony for his time today. Do you have any final comments for our audience?
No, just to say thank you to everybody for sitting in and listening. I hope you found it useful.
Thank you. I'd also like to thank the audience for joining us today and for their questions. Any questions that were submitted that we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor, Beckman Coulter, for underwriting today's educational webcast. This webcast can be viewed on demand. LabRoots will alert you when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thanks, bye.